be able to introduce Dr. Canter. I think he's a great example of someone who has worked hard from starting out as a junior faculty to thinking about how am I going to develop a research career and how am I going to figure out how to make that work with my clinical career and develop some expertise and figure out how to focus and then go through the long, arduous, and sometimes disheartening process of writing zillions of grants before one comes through, and you often want to give up hope along the way, but persistence is really the key to success in so many of these things, and often leads to great reward. And I'm really proud of Dr. Canter's now big U01 to look at the work that he's been doing in immunotherapy. He's also was ahead of the curve, I would say, in this area where at a time doing work in this kind of people thought, they've been doing this for a long time, it's never going to work. And now there's clear evidence that immunotherapy is changing the face of cancer treatment. And many of these new approaches are making a significant difference in prolonging life and the quality of life for folks. So we have a real star among us, and I'm really pleased to and proud of you and pleased to introduce you and looking forward to Of Mice, Men, and Dogs.
Great, thank you.
Thanks,
thank
Dr.
you. Clearly, I've been able to fool a few people along the way. So thank you very much. I appreciate that very kind introduction. So for those of you who've seen some of my talks before over the past few years, I tend to show a lot of pictures of my kids, for better or worse. And this is my family from last summer before the kids went back to school. There's my son doing the I love you sign from McKinley Montessori School, which I've often done in the OR, as you know. But for this talk, I'm actually going to do a little bit of a tribute to my father. He died last year at age 86, and he was a big influence on me, a big role model for me, and he was always my biggest fan. So in honor of him, I'd like to pay a little tribute to him during this talk. And this is a picture of him coaching my soccer team when I was like 6 or 7 years old. And I love these pants. They're great. And if you haven't noticed, this is me right here standing next to him with that very happy look. So why immunotherapy? I think, and why cancer immunotherapy? I think unless you've been living under a rock for the last 5 years, you have heard of how important and how breakthrough immunotherapy is, especially in cancer. And it was voted the scientific advance of the year not once, but twice, actually. Okay? And also for us, even though those of you who are not in cancer therapy and oncology, cancer is becoming more and more prevalent. Although we're doing better, there are more survivors, but we all know friend or family who has been diagnosed with cancer. And so it's important, I think, in medicine to have some familiarity with what's going on both scientifically and clinically in our patients and our friends and our family. And it really is a big deal, okay? And that's why there is a lot of hype there, okay? Immunotherapy can cure patients and advanced patients who've exhausted all other treatment options. So this is a very famous patient. And if you haven't seen the Ken Burns documentary on cancer, I highly recommend it. This is a very famous patient named Emily from University of Pennsylvania, which I have a strong connection to. And she was a young girl with a very aggressive form of leukemia, had exhausted all conventional chemotherapy treatment options, and she received experimental CAR T-cells, which was recently approved in 2017 as a new cancer therapy, and she was completely cured. And this is a picture of her, obviously a very dramatic and inspiring difference. This was at her 3-year mark, but she's now, I think, greater than 5 years out and completely disease-free. So it's a big deal. Now, for those of you who are skeptics and say, well, that's anecdotal, what is it really, behind the hype? This is one of many examples of the differences that you see. And for those of you who are not used to looking at Kaplan Meier curves, this is a big difference. When you see such a dramatic separation of the curves, it's really a meaningful effect. And this is with another type of immunotherapy, a monoclonal antibody. And the other thing that makes immunotherapy exciting is it's had success in cancers which were previously refractory to all other types of therapy. So melanoma, no chemotherapy works. Lung cancer, very limited efficacy. Bladder cancer, Merkel cell carcinoma. So there have all been immunotherapy approved for these, and sometimes more than one, for these cancers, which previously had very limited or no treatment options. And by contrast, this is the Kaplan Meier curve from erlotinib, which is a tyrosine kinase inhibitor, which was approved 10 years ago, or 9 or 10 years ago, for pancreatic cancer. It was approved based on a 10-day improvement in overall survival. So from 5.9 to 6.2, the way they presented the data make it seem like it's more than it is. 0.3 months is not even 10 days. Actually, it's 9 days. And so this was approved for pancreatic cancer. And, we can discuss sort of the, nuances of how that could happen and how our FDA works and so forth. But it's an important counterpoint to the curve I showed you before about what many people argue really is a meaningful improvement and meaningful difference in survival with these therapies. So, what are the issues? Obviously, it's not all a bed of roses, okay? And so even though it's very exciting, the reality is only about 25% of patients respond to immunotherapy. Now, like I said, those patients who do respond can have dramatic responses and even be cured, but it's still only a minority of patients who actually respond. And that's the main focus of the talk today, is sort of how to deliver the promise of immunotherapy to more patients and from the research side of things. But other important issues and barriers in immunotherapy are toxicity. I'll touch a little bit on toxicity with these treatments because you are unleashing the immune system, which can sometimes have significant side effects. And then cost, which is always an issue, and particularly in these therapies is an issue, but more of a societal issue, which I don't really touch on, but I'm happy to discuss. So this is a picture of me, I think, from junior high. And there's my dad again as the coach. I think this was an actual trophy. This was before the days of everybody gets a trophy. But I don't remember for sure. So this is, so I'm going to talk about today two of our moonshot grants that we put in last year, and kind of, the background and what we were proposing, and feel free to offer any critique that you want. So in the first one, this is more about preclinical modeling, and this is where the mice comes in. Okay, so there's two main ways, to simplify things a little bit that we, can study cancer research. And one of the big ones, and there's often sort of a spectrum from beginning to end in terms of bench to bedside. One of them is clinical trials, okay? And that's when you have something that, you feel has sufficient background data to really bring it to patients and assess if it has efficacy and safety and so forth. The issue with clinical trials is it's in some ways a very empiric or trial and error approach. And that's borne out by the fact that, a very small minority of drugs that work in mice and work in the lab actually are successful in people. And again, there's many reasons that occurs, but that's an issue that we need to address, in research and in clinical medicine, okay? And that's also, evidenced by the fact that there are about 1,000 immunotherapy trials going on currently right now, because what happens is people want to sort of, throw everything against the wall and see what sticks. All right, on the other hand, preclinical modeling, which is often mouse modeling, has a lot of significant benefits to it in terms of mechanistic studies and really looking at the immunology or the basic science, and has led to certain key advances which we could go into. But the problem is there's often a disconnect between here and here. It's easy to cure cancer in the mouse. Okay, it's easy to, or relatively easy to really dissect all of the pathways and genes in mice, but often that doesn't necessarily translate one-to-one to humans. So there's an issue there. And one of the things that we think is an issue that is worth exploring is how we actually model things in the lab. Okay, so often cancer in the lab, or frequently, and until recently, almost always, was predicated on very homogeneous, not diverse models of young mice, female. All right, the reason we often use female mice in the lab is probably like humans, they tend to fight less. Lean, inbred, and specific pathogen-free, which we'll talk about. So you often keep these mice for a variety of reasons in very germ-free environments. But that's really not how it works in the clinic, okay? And we're seeing this more and more. Cancer is a disease of the aged. It's very diverse. Our patient population is very diverse. And importantly, increasingly obese. As you may have heard, by the end of this decade, obesity is going to replace smoking as the #1 cause of cancer in the Western world. And another important point is that our patients have extensive pathogen exposure history, and that has an important impact on, especially in immunotherapy, on the immune system and on the immune repertoire. Okay, and we've seen this. This is a paper from about 5 years ago where we saw that just examining young versus old mice, this is from toxicity, there was a dramatic difference in how they responded to immunotherapy. So the aged mice all died on day 2, whereas the young mice all survived, okay? And this was linked to severe organ pathology across multiple organs and significant differences, very significant orders of magnitude differences in cytokine levels. And so this sort of got us to thinking, really, can we be doing a better job of modeling these things, especially given, how the patients we're treating in our clinic. And the other thing, obviously, again, it seems obvious, but we were exploring is aged mice, have a higher obesity content to them, okay? And this is an example, when you take older mice, they have a tendency, especially mice in the lab, which are not getting a lot of exercise, obviously, they tend to put on a lot of weight, and they tend to get more visceral adiposity. So then the question was, well, is it the obesity, or is it the aging, or is it the obesity that's playing a role in the toxicity from immunotherapy? Okay, so what we did is we took leptin knockouts, okay? And leptin is a very important hormone that helps control hunger and satiety. Okay, it's more of the satiety hormone, and obese patients tend to have actually high leptin levels and are leptin resistant. Leptin also has a lot of other important effects on the immune system and other physiological functions. But we have leptin knockouts, and these mice are extremely obese. So we wanted to take the young versus old and obesity versus lean aspect into things. And so using leptin knockouts, we saw that even the young mice would succumb very quickly and die from immunotherapy. So that indicated to us that obviously obesity is probably a critical component in the aging-related mortality from immunotherapy. And then what we did is then we said, well, given that we had seen these very high TNF levels, could we block it and could we reverse the effect? And so using TNF blockers, which are used, in rheumatoid arthritis and Crohn's disease and other diseases, using those same agents, we found that if we block TNF, we reverse many of the effects. So, again, sort of underlying this implication that age and obesity are changing, that this is changing the immune system, changing the response to these immunotherapies, and impacting both cancer progression and toxicity. So that really led us to a bigger question, is how can we incorporate these immune-modifying factors to better kind of model cancer in the lab and the response to cancer immunotherapy. So age and obesity, I mentioned, are two key factors. Sex, obviously, is an important factor. And then pathogen exposure, okay? Because your previous pathogen history and the way your body responds to pathogen exposures changes your immune system and therefore changes kind of how you respond. All right, this is my graduation from college. My dad was a huge Princetonian and very, very happy occasion. So this is where the mice and men kind of comes in. So anytime mice and men is kind of a cliche. Anytime a study comes out about mice, you'll often see that used as the appellation, but I had to use it for this just because it was easy. But it's important to know where it comes from. Okay, Of Mice and Men was a book from John Steinbeck, who was a Californian from Salinas Valley. Wrote a lot of famous books. His great American novel is considered, I think, Grapes of Wrath, which is a great movie with Henry Fonda, for those of you who haven't seen it. And his was based on a very important poem, or a very famous poem by Robert Burns, who was a Scotsman, about To a Mouse, about the best laid plans of mice and men often go awry, which is very relevant for research, for any of you who are doing research. All right, so back to the immune system. So as we know, or should know, but often forget, the immune system is highly, highly complex, okay? And I think we often, take it for granted to some degree about how incredibly complicated it is to really balance, the ability to fight off infection, keep us healthy, but avoid autoimmunity. Okay, so it's very powerful. And even within all these different cell types, there's different subsets and different pathways that they all link up, and it really becomes very dizzying in terms of trying to keep up with all that. But the important point to remember is that it's really highly, highly orchestrated and regulated. And like I said, we forget about that, right? This is, which we don't see as much anymore, but an end-stage AIDS patient, and even in our AIDS patients, they're not just dropping dead with, every time, even with a very important part of their immune system, helper T cells knocked out. Because there is so much overlap and so much redundancy, and something that's been evolving over billions of years. And these are all the things that allow our immune system to really be incredibly effective. Its ability to be specific, to tolerate the normal, self versus non-self, speed, memory, etc. Okay, so it's really highly complicated, which I think is what has made it such a challenge to study and understand. And then the other thing that's very important is how we interact with the environment, especially our immune system. And as I indicated, it's sculpted or shaped by the pathogens we're exposed to, and changes over time, and changes with aging, and changes depending on your pathogen environment, et cetera. So there's genetic and epigenetic effects that impact its work, and all of these, I said, are very important in terms of things that we can study better in terms of age, obesity, and pathogen exposure, because they modify not just in between people, but within our own immune system over time. Okay? And this is most relevant, I think, in T-cell exhaustion, and this is an important point to remember, because this is where a lot of the focus has been on T-cells, and immune exhaustion. Okay, so not to get into too granular, but a little bit of an explanation here, I think, is important, again, because this is relevant to the PD-1 inhibitors and even CAR T cells a little bit. So in a naive T cell, okay, which are matured in the thymus, okay, and the thymus involutes over age, so older people don't really have a working thymus to any degree. Naive T cells are produced in the thymus, and they're antigen specific, okay? They have a specific T cell receptor which, from genetic recombination can recognize potentially billions of different amino acid sequences, but it's specific for really only one sequence, okay? It then has to meet that antigen at some point, and if it's in the setting of a CD8 cell, it's presented on the cell surface. If it's through CD4 cells, it's through an antigen-presenting cell. But not only does it have to meet that antigen, it has to have additional signals to make sure that it's being presented and activated in the right format, okay? And that's where we talk about the 3 signals of antigen stimulation, okay? So signal 1 is meeting the antigen, signal 2 is being co-stimulated by co-receptors, and then signal 3 is often inflammation or cytokines. And that allows a T cell to mature into what we call an effector T cell. That's an armed T cell that can attack its pathogen or attack its infected cell or et cetera. And that leads to, if it's successful, to antigen clearance and resolution of the infection, and then a contraction phase, and that leads to what's called a memory T cell. And that's, again, like I said, one of the key factors of the immune system is to be able to respond better, faster, stronger to future encounters with that same antigen or pathogen in the future, all right? But what can happen is, and this is where the concept of exhaustion comes in, and it's been best worked out in a chronic infection where the inflammation and the infection is not resolved, and this T cell becomes exhausted, okay? And that's where you have these upregulation of these checkpoints that are now the focus of drug therapy to try and reverse that exhaustion. So the analogy here is like, well, you have someone who's just run a 100-yard dash, and they're exhausted, they can't really keep going, and so they shut down. The issue, and sort of the underlying rationale for this is that, in chronic infections, exhaustion has evolved to limit autoimmunity, to sort of shut some of these cells off, to prevent them from attacking self and leading to organ damage and sort of, effects on the normal host that we would want to avoid. That's the rationale. Now, it's not necessarily entirely clear if that explains everything, because you can make the argument that up here, it's the same kind of thing in terms of running a 100-yard dash. Why wouldn't these cells be exhausted too? And everything kind of happens in nature for a reason. So why are we upregulating these molecules, these checkpoints, to shut them off on purpose? There's more to the story there, but that's the way people talk about it and try and explain it, okay? And that's why we have exhaustion. So how does this work in cancer? In cancer, it seems like what happens is we have T cells. We have T cells homing to the cancer. The cancer have new antigens because they're highly mutant. And a lot of DNA damage, they express neoantigens, which are different from self enough that the T cells recognize them. And then the tumors then upregulate PD-L1. PD-1 and PD-L1 stand for programmed death ligand and programmed death receptor. Again, these are there for a reason. It's not just an accident, right? And these induce apoptosis and shut these cells down. So what happens a lot in cancer is then the tumors upregulate PD-1 because they're shutting down the responding T cells, okay? And this leads to exhaustion. And this is important because these are 2 of the important factors that predict response to PD-1 inhibitors, okay? Tumors that have a lot of neoantigens or have a high mutational burden, and tumors that express PD-L1, those are biomarkers of patients who are going to respond better to checkpoint inhibitors, all right? And this is what we call a T cell inflamed or a hot tumor, okay? Whereas there are other tumors that are totally cold. And again, is it because of the mutational burden and the expression of neoantigens, or is it because of other factors? And that's obviously an important area of research in terms of identifying these immunosuppressive pathways, et cetera. But this is sort of the classic model, and this is where we know there's an effect from PD-1 inhibitors, okay? But the issue is what kind of dictates kind of these expression of exhaustion markers, and that's something that we're looking at. And one thing that we found is that immune exhaustion increases with age and obesity. So it's not just in cancer and expression of neoantigens. These pathways and these signatures are impacted by other immune modifying factors. So we've seen in mice as they age and as they're older and as they're more obese, they express PD-L1. For those of you who are not used to looking at flow cytometry plots, we're detecting the expression of these proteins, cell surface markers, because they fluoresce a fluorescent light, which we can detect by a laser. Okay, so here is PD-L1 on the x-axis, and here is on the y-axis just our marker to kind of gate around different cells. Okay, and these are CD4 and CD8. And the point here is, like I said, is with obesity you're having greater expression of exhaustion markers. This is mice. This is humans. Okay. And then similarly, or related, is we found that with the microbiome can be impacting these things as well. So just sort of take a little bit of an off-road here. The microbiome, as you may or may not know, is the collection of all the organisms that are sort of living in and around and on your body. It's a very hot topic of research right now. And again, for those of you who don't know, your microbiome far exceeds your actual own cells in and around your body, okay? So, by 10 to 100 to 1. You have more microbes living on you and in you than your own cells, all right? And the microbiome has been implicated in many aspects of health and disease, all right? And, dogs and their owners actually share the same microbiome. I mean, it's a very fascinating kind of thing, all right? But it also has been shown, interestingly, to affect your response to immunotherapy. All right, and is it because of the different microbes in your body, are sculpting your immune system, like I mentioned, okay? So we found also that your diversity, and it seems again like the diversity of your microbiome is a key factor, that the diversity of your microbiome decreases with obesity and age a little bit, okay? So this is another area which is relevant to aging and obesity, all right? And then finally, your infectious history impacts your immune phenotype. So we've seen how in viral models where you give MCMV, which is analogous to CMV, a ubiquitous virus, that obese mice have more exhaustion and less ability to clear the virus when exposed to MCMV. Okay, so here, quantifying the number of exhausted cells by expression of PD-1, lean and obese at an early time point both have exhausted T cells, but then the lean are able to clear it and return to homeostasis by day 18, as you would expect, whereas the obese mice remain exhausted. And this probably induces a refractory period in terms of their ability to fight infection. But the implication of this, we hypothesize, is that obese people may respond better to immunotherapy, interestingly, because if they have more exhausted cells, T cells, and we know that PD-1 expression is a marker for response to immunotherapy, our hypothesis is that obese patients may respond better. And it's something that no one's really looked at as yet in the clinical experience, but it's worth evaluating. The problem is that the obesity itself is a cofactor which can change the tumor kinetics. Okay, so as I mentioned, obesity is a cause of cancer or a contributing factor to cancer clinically, and certainly we've seen and others have seen that obese mice, the tumors grow faster. Okay, so here DIO is diet-induced obesity. When we make our mice fat, which can take 6 months of feeding them a high-fat diet, and the type of diet you give them is relevant as well. Some people give them what's called the cafeteria diet, which is high in sucrose and fructose. We give a high-fat diet. There's different ways which may be relevant. But regardless, our obese mice, the tumors grow fast. Okay? So it's a double-edged sword. And here's a mouse PET scan, which I thought was kind of cool. you can do some pretty cool small animal imaging. The tumors are more metabolically active. So, that doesn't necessarily change the observation that obese patients may respond to immunotherapy better, but it makes it harder to study because, we're fighting a faster-growing tumor with the tumor we're trying to treat. But yet we've seen that the DIO mice do seem to respond better to therapy. So there's enhanced anti-tumor effects in obese mice, which, like I said, may be clinically relevant. All right? And we're intending to study this long term because that's another problem with mouse modeling is the xenogenic issues. So you can't necessarily study long term like you would want to in people. And then finally, as I mentioned, your pathogen history is very important. A lot of research with mice is on what's called SPF, or specific pathogen-free mice. You have these biosafety level requirements, and you want to keep them germ-free to prevent spread of disease and so forth. A lot of investigators are working with immunosuppressed mice, and this actually led to a very impactful paper where these researchers in Minnesota were actually catching wild mice, which was a whole other level of IACUC regulations. But they were catching wild mice and studying their immune system and found out how tremendously different the wild mice immune system is from our laboratory mice, which has key implications for what we're studying. Maybe a lot of the things we're observing are artifactual because it's not really how things are working in the real world, so to speak. But we've seen that our pathogen-free mice have an immature immune system, okay? So they're not exposed to the same number of pathogens, either acute or subacute, and this leads them to have a very restricted and potentially less able to respond to pathogens over time, because as you age, you lose some of your ability for immune system to respond to new pathogens, because of thymic involution and et cetera. Mammals were evolved to really have a very active immune system up till the age of reproduction. And so, as we know, older patients are more immunosuppressed, but by limiting their exposure, we're immunosuppressing them even more. And this is where you should learn about things like the hygiene hypothesis, and why people who are not exposed to things and younger, all the alcohol, gels around, why there may be more allergies around, okay? Because peanut allergies and so forth. The immune system seems to need a certain amount of stimulation to really age and mature appropriately. So we've seen that with our SPF mice, that they have an immature immune system. And interestingly, when we co-house them with mice you can buy in the store, so you can buy, there's laboratory mice, there's mice, pet mice, you can buy like PetSmart mice, and then there's wild feral mice that you can trap in barns and stuff. Okay, so when you co-house them, which probably allows for a commingling of different subacute infections, mites and so forth, it also impacts their immune system. So all of this we sort of combined into our moonshot grant to really kind of study all of these factors and how they could make a more relevant mouse model of cancer. Okay, so we had our obese mice, our young and old mice, our pet store mice. We wanted to look at immune phenotypes, metabolic profile. We wanted to look at exposure to different pathogens, being MCMV, which is a model of chronic infection, influenza, which is a model of acute infection, add tumors in, add immunotherapy, and kind of look at how all these things played out, in time and space. This one didn't get funded. I won't give you all the details, but basically it was too complicated. And so that was the sum of the review. It was hard to know what all the different things were working together. And there was, not a mechanistic point of view to look at all the different ways these factors were interacting. Okay, this is a picture of my dad and me when we were in Italy. I think I was college studying abroad. This is Piazza San Marco in Venice. So this led us, or not necessarily, I think we submitted this one before the other one, but this is our second moonshot grant, which was more about multi-species evaluation of immunotherapy. And why do we like, multi-species evaluation? Number one, it actually plays into our strengths here at UC Davis with our vet school, with our primate center. And number two is that we think it can speed sort of and bridge the bench to bedside translation, okay? So, and I'll talk about it a little bit, especially in dogs. It really allows us to do things faster and with decreased risk to human patients. We're treating our dog patients, but I think there's a little bit more of a willingness to do experimental therapies in dogs and do it quicker. And that's, to really kind of predict better, to avoid a lot of this sort of trial and error approach, really predict better which therapies are going to really work and which won't, or which might be toxic and therefore shouldn't be advanced, okay? And for this, we're really looking at NK cells, natural killer cells. We're one of the few labs in the world that are looking at dog NK cells. And they're a sort of, sidebar of the immune system that we've probably all heard about in medical school, but really seem to have a lot of weird properties, and hence the name. But they seem to really target virally infected and possibly malignant transformed cells and eliminate them, okay? And we've been able to grow up, like I said, dog NK cells in the lab. We've seen that radiation of targets makes them kill better, and we've been exploring how they might be a cancer therapeutic, especially because dogs actually get a lot of cancer, believe it or not, especially sarcomas, but they get melanoma, they get bladder cancer, pretty much get everything that humans get except for breast cancer because they're spayed, and they don't get pancreatic cancer for some reason. And cancer in dogs is very, very similar to the way it is in humans. Okay, it evolves in the same environment, the genetics are the same, and their immune systems are intact, so the interaction between the immune system and the cancer development over time is the same. So we, a few years ago, started a clinical trial in dogs where we were giving them radiation, which they get as palliation because a lot of them don't get amputated, and then natural killer infusions. And we actually just published our experience. This was the first in dog ever natural killer cell immunotherapy trial. And like I said, we've been interested in dogs for a while because dogs get spontaneous cancers just like humans do. They're pretty much, in many, many ways, it's very, very similar to people. The time course, the environmental and genetic oncogenesis. There are some differences in how they metabolize drugs and so forth, but like I said, comparable to people, probably more so than mice. Okay, this is when I was in med school. I came and I did a sort of pseudo away rotation with my dad. He was a general surgeon. I don't know if I mentioned that. He was a general surgeon, and I did an away rotation with him when I was a fourth-year medical student. So there is a history of doing immunotherapy in dogs, and this was done. Our proposal is first in dog interleukin-15. The history is with inhaled human IL-2, and these are cytokines. Like I said, cytokines are key regulators of the immune system. And for example, the febrile response of fevers, chills, and sweats is all modulated by cytokines when you get viral infection or so forth. So people have tried to use or have used human IL-2 as a nebulizer in dogs, and there have been some responses. You got to get used to looking at a dog X-ray here. So these are tumors that with inhaled IL-2 there is some shrinkage and some response. All right, the problem with IL-2 is it induces regulatory T cells, so it has immunosuppressive effects. So our proposal was, and one of our aims was to use IL-15 instead, because it's more of a specific survival factor for NK cells and memory T cells. So part of our proposal was to do a phase 1 trial with IL-15, looking at toxicity and efficacy. But then we want to combine it with NK cells based on our experience using NK cells. Now we think if we also provide cytokine to the dogs after we infuse the NK cells, that'll keep them alive longer and make them work better. Okay, so this is our phase 2 trial. This is sort of our complicated schema. While we're growing up the NK cells, we're then giving them inhaled IL-15, and then we're reinfusing our NK cells and looking at how they respond. And this is in dogs with sarcomas and melanomas. And then importantly, we're going to look at some of the scientific correlative studies looking at how the NK cells are homing to the tumors, how long they're lasting, and how much we can detect that in the blood in the tumor. All right, and we've seen that we can recover these NK cells in our previous trial. We can recover them after they've been infused. So here was the schema of the previous trial where we're radiating, we're growing up the NKs, we're re-infusing them, and then we can tag them and do a biopsy. So here's our positive and negative control, and this is our tagging dye, and we can see that we're recovering, the only cells that are going to have the dye are the cells we've tagged. And so when we do the biopsy, we can recover our cells 1 week later, showing that there is some effect. So, and then finally, we're going to be doing with a superagonist, an IL-15 synthetic that lasts longer and may be more potent. We're going to be doing a superagonist clinical trial, looking, at a different phase of disease, and if we can prime the immune system and have an effect at eliminating micrometastatic disease and preventing metastasis. All right, and here we're again, sort of circling back to the concept of cold and hot tumors. Dog sarcomas, similar to human sarcomas, tend to be cold. That means they don't have a lot of infiltration of T cells and NK cells. This is some of our data. So our hypothesis is that this superagonist is going to convert cold tumors to hot tumors because we're going to do amputation. We're going to have pre and post tissue biopsies, to look at these things. And then we're also planning on doing next generation sequencing. I don't understand any of these, next generation sequencing plots, but we're hoping to have some in one of our papers, okay? That's what Alicia is for. She's going to be doing some high-level bioinformatics, all right? So, this is sort of a summary. I like this picture of Sarah and Dr. Michael Kent. He's one of our vets, collaborators at the vet school. This was one of our first patients. The dog model is a great model to advance immunotherapy, especially because, another limitation of mice is really modeling the immune system because either using mouse cancer in immunocompetent mice, and mouse cancers are very, very artifactual, or using human cancers in immunosuppressed mice, so you're losing that interaction with the immune system. So the dog is a great bridge to speed translation and to optimize safety and efficacy. So this grant actually got funded, which was great, and this was some of our feedback. It's never, there's always got to be some negative feedback. It's the feedback sandwich, right? But I think they liked it, and that's why we got funded. Okay. So in summary, all right, immunotherapy is the future. I think that's important for everybody to remember. UCD can be a unique leader in cross-species studies, and innovative models are needed. That was one of the calls of these NIH moonshots, was really to get better models for bridging bench-to-bedside research. Okay, and we have current NIH funding studying novel NK cytokine strategies in dogs with natural cancers. Okay, and then give your dad a call. Well, I think that's a great question. I think there's an important role for surgeons generally in translational research. because of these window of opportunity trials, which is like our, the superagonist, where you can really assess pre and post. And so neoadjuvant kind of window of opportunity where you're giving an agent and it's an earlier stage of disease. They may not be as heavily pretreated, but you really are able to really answer a correlative scientific question and at least, get out of the empiric kind of throw a new agent and see what happens and look at very, sort of gross endpoints of, response or progression-free survival. And in addition, because, the pre and post is key for, improving your sort of statistical power, because there's so much heterogeneity, as I was kind of mentioning, between patients. Looking at the same patient pre and post really allows much more statistical power and smaller sample size and whatever to really answer some questions. But the key, I think, surgeons unfortunately get a bad rep in research, and I think there's a lot of bias against them. But you really have to, as Garth says, I got to credit him, you really have to have a well-developed question so that you can really try and come up with some not ambiguous answer, which will be meaningful. But so I think surgeons have a critical role, and I think we need to be more involved. We tend to get, there's so much bureaucracy and inefficiency, and we're all busy and we're all kind of impatient, and I think we just kind of like give up a little bit, and that's why sometimes we're less successful in research. And I think it's that's a key. It's just really got to be persistent and not get beaten down by all of the inefficiencies that slow things down. We won't result immediately.
It's a great talk. Thanks for giving it. I think immunotherapy clearly is a key for cancer therapy. My question is, and forgive me a bit naive in terms of the immune system, but it seems to me that taking, maybe you can explain what happens once you take the cells out of the patient, but you take autologous cells in a patient that has cancer, and those cells have now hung out, and they're sort of the guardians that have allowed this cancer to grow. And you take them out of the patient, and you either expand that clonal line or expose it to other antigens. How do you get them to then actually fight the cancer that they ignored earlier? Is it really, for natural killer cells, is it a numbers game, or is it change the cells in vitro before you give them back?
That's a good question. So it depends on which cells you're using. So the classic one are the CAR T cells. Those are chimeric antigen receptor T cells. Those are genetically engineered to respond to a specific antigen. So they've been transfected with a virus to express the antigen. The T cell receptor will specifically target that antigen. And then in addition, there's second and third generation modifications, which then give them co-stimulatory factors to really, juice them up. Okay, so that's why they work in CAR T cells. And the problem, actually, although it hasn't really been a major, or it's been managed, but the problem is they're so armed, they're so juiced up that they can cause collateral damage. So that patient Emily, again, very interesting sidebar. She almost died. She got the cells infused, and because they're so juiced up, they had what's called a cytokine release syndrome. And so they get, hypotensive, yada, yada, yada. And she almost died. And what's really fascinating about that case is the PI on that study, his daughter had rheumatoid arthritis. And he was just aware that, IL-6 can be a mediator of cytokine release syndrome. And he was like, give her this IL-6 blocker, which they use for juvenile rheumatoid arthritis. And that is credited with saving that patient's life. And so people have said, if that patient had died, that could have totally changed how immunotherapy evolved. So that's the way it works in CAR T cells. It's something similar in NK cells. We are, giving them, optimal conditions to grow, to be perfectly activated and expanded and cytotoxic, which they probably don't see in the body. So it's not that they're, they're being immunosuppressed and we're, on the opposite end, activating them. the problem with NK cells is they're not antigen specific, and what happens is when you reinfuse them, you take away that, they're like addicted to this like perfect, ex vivo environment, and they tend to sort of, crap out when we infuse them back in. And so that's why we're looking at IL-15 to kind of recreate, in vivo what we're given to them ex vivo. So, but those are some of the issues. Yeah, Mike.
Great talk, Bob. I have kind of a simple question, I guess. Looking at obesity and exhaustion, if the obese patient is more likely to have exhausted T cells, if you make that person thin, do those T cells bounce back? And if so, what is kind of the mechanism underlying it? Is it adipocyte? Is it endometabolic? What is causing this kind of fluctuation in the immune system?
Yeah, that's a great question, and there's not a lot of good studies on that, but one of the big areas is where did Dr. Ali go? He left. He snuck out. Is actually looking at the bariatric patients and seeing what happens to their immune system post-bariatric surgery, and that's a problem with a lot of the human studies. Is as you know or may know, it's descriptive. It's not mechanistic, and so you get conflicting results. It's hard to know. What's the real story there? But it seems like a lot of it is reversed, but not all of it. And it's so complicated. The mechanism is another great question. Again, as I tell the residents, it's multifactorial. That's always this pat answer. Leptin, our data seem to implicate leptin, but it's probably more than just one thing. But leptin is a big one. Well, in the dogs, we like the dogs. We don't like the dogs, but the dogs have melanoma and sarcoma is one we're studying in dogs because they have a lot of advanced disease, get a lot of lung metastasis, and it seems like it's an unmet need. Although melanoma we have therapies for, but and I like that because of sarcomas, obviously. That's a great question. We're hoping to be able to bring this to people at some point. The issue with bringing NK cells to people is there's just all these huge GMP kind of issues and regulatory issues, and there's cost issues. The CAR T cells, which are very, very successful, cost over $500,000 per patient. So, that is obviously an issue that needs to be addressed down the line. But, we're also looking at it as, only 25% of patients respond. Can we find, a way to help those 75% of patients who don't? But that's down the road. You can only worry about one thing at a time. Yes, sir.
I enjoyed that too. Great lecture. Does T cell exhaustion occur at the local level? I'm thinking of Marjolain's ulcer, and the explanation we all grew up with was that it was inflammation and oxidative stress, but T cell exhaustion and PD-1 expression may play a big role. And,
yeah.
That may actually be a model if that occurs.
Yeah. No, I think that's an important thing that, now that a lot of attention has been, as Dr. Farmer mentioned, immunotherapy was kind of a backwater for many, many years, which is a whole other interesting story. But the impact of the immune system on cancer formation is a really important area. And, there's a very important paper called The Hallmarks of Cancer, which describes a lot of, the hallmarks, which was mostly focusing on the tumor. But now we've realized, and now that's why there's a Hallmarks 2.0, is the host, okay? And so that's where the concept of the 3 E's of immunoediting come into play. And the immune system does have an important role in what we call cancer surveillance, which was first proposed by Lewis Thomas, 70 years ago. Which is, again, is paradoxical because, in the early stages, the immune system eliminates transformed cells. Okay, so there's cancer cells floating around that the immune system recognizes as cancer and eliminates them. But what happens is that, like everything, puts selection pressure on the cancer cells to evolve to escape that immune elimination. And that leads to sort of an immune equilibrium, where you have cancer cells and some of them are being eliminated, but there's still a nidus there. And that puts further pressure then for immune escape. And so you have cells, so the pressure on cancer cells is to evolve to escape the immune system and to proliferate and, unfortunately, become problematic for the host. So it's certainly an element of it. Probably the nidus of what's stimulating oncogenesis is still related to inflammation and evolution of mutant clones in the setting of inflammation. But the immune impacts, there's immunosuppressive effects too from a lot of those stimulatory cytokines, which can be immunosuppressive on the local effector T cell, NK cell populations. Great.